Hello, everyone. My name is Nick Chen. I'm the principal scientist of Genomic Epidemiology at the Virginia Division of Consolidated Laboratory Services. And in this section, I'll be providing an overview of pathogen diversity, how it arises, and its connections to genomic epidemiology. The goals for this section are to describe pathogen diversity at the genome level, understand sources of genetic diversity, differentiate between genotype and phenotype, and demonstrate real-world examples of pathogen diversity. Before jumping into the material, I'd like to start by saying that there are many different ways of understanding pathogen diversity. There's the pathogen disease model that may be favored in more clinical settings, the taxonomical model often employed in phylogenetic approaches, and the applied framework of genomic epidemiology. Each of these perspectives is valid, and I'll be transitioning between all of them throughout this discussion. To start, we'll take a look at different groups of pathogens and their genetic characteristics. If we adopt a disease-oriented perspective, we can roughly divide up pathogens into one of four groups: bacteria, viruses, fungi, and parasites. For each of these, I've listed some common examples of pathogenic agents, the diseases they cause, and a common treatment approach. It should be noted that these four groupings can be subdivided many times over, but this rough division provides a good entry point into understanding their genomic characteristics. Beginning with viruses, these are neither prokaryotic nor eukaryotic, and typically have very small genomes on the order of about 3 to 200,000 nucleotides, which are the building blocks of DNA and RNA. These genomes are very efficient in that the vast majority of the genome encodes for some type of protein. If we look at the figure on the right, we can see that the structure of the viral genomes is quite diverse, being comprised of DNA or RNA in various structural configurations. Finally, due in part to their small and variable genomes, viruses tend to be quite diverse and it can exist in what are referred to as quasi-species, which is a population structure of closely related yet variant genomes related by some mutations. Moving on to bacteria, these organisms are prokaryotic, meaning they lack a nucleus or other membrane-bound organelles. And compared to viruses, their genomes are considerably larger, on average ranging from about 2 to 10 million nucleotides. Generally, the genome is a double-stranded DNA arranged in a single circular chromosome, although there are some exceptions to this. Additionally, if we look to the figure on the right, we can see that the genome has two distinct components. The core genome is comprised of 3 to 5,000 genes on average that are present in most strains of a given species, while the accessory genome is comprised of thousands of genes that may or may not be present in the genomes of individual bacteria. Taken together, these two components constitute the pan-genome. Finally, we can look at the genomes of both fungi and parasites under the umbrella of eukaryotes, which contain both a nucleus and other membrane-bound organelles. These organisms have significantly larger genomes than both bacteria and viruses, oftentimes being comprised of billions of nucleotides, as is the case with the human genome. These genomes are arranged into multiple chromosomes of double-stranded DNA and have significant regions that serve no apparent function. These regions, referred to as intragenic regions or introns, are spliced out as a part of the post-transcriptional modifications and thus are never translated into proteins. Now that we've looked at the genomic characteristics of these broad pathogen groups, it's worth talking about how this genetic diversity arises. To do so, we need to look back at the central dogma of molecular biology. The central dogma holds that DNA is transcribed into RNA, and this RNA is then translated into polypeptides. If we take more of an epidemiological perspective, we can then say that a single organism replicates into multiple organisms, and these organisms are transmitted, leading to an outbreak. Now, each of these stages, there is a potential for genetic diversity to be introduced, and we'll get into many of these mechanisms in the following slides. Before doing so, however, it's necessary to revisit the idea of the genotype and the phenotype. Broadly speaking, the phenotype of an organism is its observable traits. In bacteria, these may be antibiotic resistance characteristics, the peptidoglycan composition of cell walls, or their overall morphology. This is distinct from the genotype of organisms, which relates to its genetic code. This genetic code may be conceptualized in various ways, from single nucleotides to whole genes and their alleles. Finally, there is the environment, which can be thought of as the factors that an organism is exposed to, such as a localized climate, the availability of nutrients, or the presence of harmful chemicals. While it's often thought that there is a one-to-one connection between phenotype and genotype, the relationship is somewhat more complex due to the influence of environmental factors. For example, a bacterium may possess an antibiotic resistance gene, but if there are no antibiotics in the surrounding environment, it may not express this gene due to the metabolic cost it incurs. On the other hand, another bacterium may lack this specific resistance gene, yet can demonstrate a degree of resistance to the same antibiotic through other mechanisms, such as efflux pumps. While the relationship between phenotype and genotype is complex and influenced by many factors, we can broadly say that the observable traits of an organism are dictated by a combination of its genetic code and the context in which the organism exists. Now, to begin looking at sources of genetic diversity, we'll start with mutations. These are caused by external damage or errors in replication and may be beneficial, neutral, or deleterious to the survival of an organism, although most tend to be neutral. The smallest form of mutation is a single nucleotide polymorphism, or SNP. You also may see these referred to as single nucleotide variants, or SNVs. However, there's a slight difference between these two terms. SNPs are caused by errors in DNA replication or RNA replication, RNA viruses, and constitute a change to a single nucleotide. If we look to the figure in the top right, we can see three such SNPs, wherein the three nucleotides in blue are substituted with the nucleotides in red. These changes are affected by the accuracy of DNA or RNA polymerase, as well as the cellular repair mechanisms, and make up the smallest form of genetic change detectable when comparing organisms through whole genome sequencing. These changes can then lead to what are called synonymous or non-synonymous changes. Synonymous changes are when a SNP does not lead to a change in the amino acid. We can see this in the left-hand portion of the figure in the bottom right, wherein the starting codon, or triplet of nucleotides, codes for a leucine amino acid. If we change the A nucleotide to a G, this codon still encodes for a leucine amino acid. This would be a case of the genotype changing, but the phenotype remaining unchanged. Now, if we look at the right-hand figure, we will still start with a leucine, but now if we change the A to a C, the codon encodes for a phenylalanine amino acid. This would constitute a non-synonymous mutation and would be an instance where a change to the genotype does lead to a change in the phenotype. Finally, if we compare the ratio of non-synonymous mutations to synonymous mutations in a so-called DN/DS ratio, we can get a better understanding of the selective pressures the population is currently facing. When discussing mutations, it's important to note that not all organisms have the same rate of mutation. Typically, we see a correlation between increasing genome size and decreasing mutation rate, although this relationship is neither linear nor universal. Moving on to another type of mutation, we have insertions and deletions, often discussed under the blanket term of indels. Indels are distinct from SNPs because whereas SNPs are substituting one nucleotide for another, indels entail adding in or removing nucleotides from the genome of an organism. Because codons are comprised of 3 amino acids, we often see indels in units of 3. Otherwise, they may induce what is called a frameshift mutation. Aside from mutations, there are many ways genetic diversity can be introduced to a population. One such method is recombination, which is the exchange of genetic material between chromosomes or between different regions of the same chromosome. This process looks very different depending on the specific organism. In humans and nearly all eukaryotes, recombination entails a crossing over of sister chromatids, as we see in the figure in the top right. In viruses, this process requires co-infection of the same host cell, as we see in the figure in the bottom right. And in bacteria, there are several distinct processes that we'll discuss more on the next slide. In addition to recombination, there is a process specific to segmented RNA viruses referred to as reassortment. This is similar to viral recombination, except rather than swapping non-specific lengths of genetic material, whole segments of the genome are swapped. This process is known as genetic shift in influenza, whereas the accumulation of diversity due to mutations is typically referred to as genetic drift. Focusing now on bacteria, these organisms have three distinct ways of exchanging genetic material, as we see in the figure on the right. In conjugation, genetic material is directly transferred between two living bacteria. In transformation, free genetic material is taken up from the environment around the bacterium. And finally, in transduction, genetic material is transmitted from one bacterium to another through a bacteriophage, which is a type of virus that targets bacteria. Each of these horizontal gene transfer mechanisms relies on easily exchangeable genetic materials known as mobile genetic elements or selfish genetic elements. One of the better known types of mobile genetic elements are plasmids, which are circular double-stranded DNA structures that can replicate independently from chromosomes, often carry resistance or virulence genes. It can be passed from one bacterium to another. In addition to plasmids, mobile genetic elements can be in the form of insertion sequences, transposons, integrons, or cassettes, all of which play an important role in transferring resistance and virulence genes. Up until this point, we've been discussing genetic diversity at the level of individual organisms or in small populations. However, there are important drivers of genetic diversity at the more epidemiologically relevant level of individual patients. If we start with a single patient or host, represented with the circle on the left, we know that diversity will gradually accumulate in the pathogen population due to the factors that we've been discussing. In a single host, many pathogenic variants, represented by the various colors on the left, will arise over time. When this individual transmits their infection to another, say through a sneeze, only a small fraction of the total pathogen load is being transmitted. Further, this small fraction is more or less a random sampling of the genetic diversity of the host. In this case, we see that host 1 has predominantly orange variants, while by random chance, the inoculum in the middle is comprised of 2 red and 1 blue variants. This random subsampling is referred to as a bottleneck because it only allows for small representation of the overall diversity to be conveyed. Now, once this inoculum infects another host, it will begin to grow and diversify within this new environment. While again we see new variants arise, the composition in host 2, represented by the circle on the far right, is still mostly reflective of the inoculum with mostly red and blue variants. This is what is referred to as a founder effect, wherein the population that develops in a new environment is largely reflective of the founding population. The amount of diversity in the overall population is strongly influenced by the degree of diversity in the founding population. In turn, the degree of diversity in the founding population, or the inoculum in this case, is affected by the degree of diversity in the population of host 1, as well as the size of the bottleneck. Therefore, we could think that a quickly mutating virus that requires a small infectious dose may accumulate much more diversity through a transmission chain than, say, a bacteria that mutates slowly and requires a much larger infectious dose. Often, when we are conducting epidemiological investigations, we are working in calendar time, whether it be time from infection to symptom onset, from one case to another, or from the onset of an outbreak to its resolution. This is what our next topic, molecular clocks, is so important to the practice of genomic epidemiology. Molecular clocks are simply the quantification of the evolutionary rate of an organism. It's important to note that an evolutionary rate is distinct from the mutation or the substitution rate in that it's influenced by both internal as well as external factors. This process of quantifying the evolutionary rate is so important because it allows us to convert measurable mutations into calendar time. For instance, if we know that an organism mutates at a rate of 12 SNPs per year, we can expect to see approximately a 12 SNP difference between 2 samples taken a year apart. Translating this to epidemiology, If we know that the average time it takes for a certain pathogen to be transmitted from one person to another is a month, and we know the evolutionary rate is, say, 12 SNPs a year, or about 1 SNP per month, we should expect to see 1 SNP difference between samples taken from directly related cases. If instead we find that 2 patients have samples that are dozens of SNPs apart, it's very unlikely that one case directly infected the other. It should be noted, however, that molecular clocks are not a static attribute of pathogens. Oftentimes, we calculate these evolutionary rates by fitting linear regression to a large set of isolates gathered over a fixed period of time, as we can see with the black line in the figure on the right. As we can see in this figure, however, the black line is a better fit in some locations than others. Some data points fall above the line, indicating a higher evolutionary rate, while others fall below it, indicating a slower evolutionary rate. This is a limitation of these so-called strict molecular clocks, and where a relaxed clock model may be a more appropriate fit. Relaxed clocks allow for changes over time, potentially allowing for a more accurate calculation at the cost of greater complexity. Another consideration is how to express the clock's rate, for which we have 2 options. If we adopt a normalized rate, we express this value as the number of substitutions or mutations per site per year. In other words, an organism where we expect to see 100 substitutions that has a genome length of 1,000 bases would yield a normalized rate of 0.1 substitutions per site per year. This is great for comparing between organisms because we are essentially standardizing the rate. Now, if we adopt a non-normalized rate, we'd express this value as a number of substitutions across the entire genome per year. Using the same example organism as before, our non-normalized rate would be 100 substitutions per genome per year, whereas before it was per site per year. Now, this is far more easily understood as we are often working with a single organism and may not know the expected length of its genome. Both of these approaches have their uses, and converting from a non-normalized rate to a normalized rate is as simple as dividing the non-normalized rate by the length of the genome. Pulling all of this together, if we look at the figure on the right, we can first see that this is a collection of SARS-CoV-2 genomes with collection years on the x-axis and the number of accumulated mutations on the y-axis. Because there is only one regression line and one corresponding rate, we know that this is a strict clock model. Additionally, when we look at the rate, it may not be immediately obvious that this is a normalized or non-normalized value. However, if we look at the regression line and see where it would intercept the y-axis after a year, this value is just about 30. Therefore, we know that the rate is not being divided by the length of the genome. Otherwise, this value would be closer to about 0.001. Now, I'd like to briefly touch on a few examples of pathogen diversity in the real world and how it interfaces with epidemiology and public health more broadly. Starting off, while there is much to say about the COVID-19 pandemic, one of the more notable aspects of it is how it introduced the term viral variant to the public lexicon. Whether through the more technical nomenclature system seen in the figure in the top right, or the more common names of Delta, Omicron, and so forth, these variants demonstrated how genetic diversity can accumulate and its impacts on public health. When understanding the evolution of this pathogen, it's important to note that the mutations we see accumulate over time were not randomly dispersed throughout the genome of the virus. Instead, we saw a concentration of mutations in the S gene region of the genome, which encodes for the spike protein. This is clear in the figure in the bottom right, where we see far more mutations and a much higher frequency of these mutations, represented by the black lines, in this S gene region of the genome relative to other gene regions. This is because the spike protein plays an important role in the ability for the virus to infect new hosts and therefore to spread and to multiply. Therefore, there's a strong evolutionary pressure to select for particular phenotypic changes in this region, leading to increased genotypic diversity. And this genotypic diversity had far-reaching implications for public health, ranging from molecular testing, the efficacy of antibody-based treatments, the formulation of vaccines, individual health outcomes, and epidemiologic practice more broadly. In turn, the way we reacted to these impacts of genetic diversity also influenced the selective pressures driving the genetic diversity to begin with. We can think of this in the context of vaccinations, where a higher coverage of vaccination in the population creates a selective pressure for variants with higher immune escape potential. Another example of pathogen diversity is the recurring, emerging, and persisting bacterial strains often associated with foodborne outbreaks and tracked by the PulseNet system. These organisms may repeatedly cause acute outbreaks in the case of reoccurring strains. They may be novel or rare strains that increasingly cause illness or have the potential to, or they cause illnesses consistently over a long period of time. Whereas with SARS-CoV-2, we saw a single virus that diversified into variants with distinct phenotypic and epidemiologic characteristics. Here we have a diverse set of organisms, including Salmonella, E. coli 0157, Listeria, and Campylobacter, that are associated with similar etiologies, transmission routes, and symptomologies. In summary, there are many different valid perspectives on genetic diversity. Pathogens have highly varied genotypes and phenotypes. There are many distinct mechanisms by which genetic diversity arises. And understanding how, where, when, and why genetic diversity occurs is a key component of genomic epidemiology.